Hey guys, since there are only three of us, I guess if I start a minute earlier, nothing will happen. What's happening with everyone? Is it exam time? I see less and less people every day. What's going on?
I think finals have started for undergrads, if I'm not mistaken, by today.
Today? Oh, probably. I mean, I'm not even aware of exam times, but doesn't matter. I'm uploading it online. And guess can you access review session online? Yeah. Okay. Good.
There also is another link right now too that I clicked on that didn't have like it was the first one when I like signed on to the Zoom things. It says that there is one reoccurring and the meeting idea is 922-8231-1625. So a lot of people could be potentially there instead of 983.
So let me see. Say the link where say that link again.
It's
The number.
the idea is 922-8231-1625.
So that's not the one. Wait a second. So is that the one that I should be maybe, right?
I think that is the one for Lisa's, but then there's one today. So I don't know. Maybe people accidentally clicked on that one and they'll back up on this one.
Yeah, but I don't think I can. Wait. Let me see. Maybe it's there. Let me check. Because that one is like I cannot record there because it's Elisa is host. So
yeah.
Let's see. I will try. Can I log into other link also? Right? I think I can.
I think so, maybe. Or maybe now people are coming.
Let's see. I will try to. Let's see. So I think now I will probably leave meeting and then I will come back.
Okay.
Nobody was there.
Oh.
You can't actually. Wait a second. Can you guys hear me? Because okay, it's fine. Because my computer says I'm muted and I'm not. Can you hear me?
I was like waiting in the top because there's like the two links and it said I was waiting.
Huh.
Yeah, that's where I said Marina too, because I was like, there was two different meetings. So
yeah,
like maybe over there.
But I mean, people will figure it out.
I think I know what I did. I have one link that I saved in my calendar, and I have another one that I saved in e-class, and I logged in both of them. But now we are good. Anyway, let's get started because I'm scared that today we will really have a lot of work to go through. So if I am rushing a little bit, I apologize. That's just because first of all, Unit 7 is complex. Second of all, Unit 7 is very significant for your exam. So Unit 7 is a key point. And I know that because I saw exam. So I want to go through Unit 6 also, but I will go through Unit 7 in more details. Again, no freaking out. If we are going too fast, that's all fine. All these notes are uploaded online, so you can see everything. So I'm sharing a screen now for the Unit 6, and we are going through that right now. Okay. So Unit 6 is bioenergetic. That's a part that I considered easy, just because that's what I am studying my whole life. And it's a general microbiology knowledge. So this is important to understand because if you don't understand this, you won't understand anything from here. So this is kind of key point, all metabolic pathways. So again, I will not read all of that just for the sake of time. What I want you to bring with you from this class is that prokaryotes are chemists of the world, so microbes. These definitions, lithotrophs, organotrophs, and phototrophs, are important for your general microbiology culture. So you won't be asked on exam to define a lithotroph, organotroph, or prototroph, but in future, you do want to know that if something is chemolithotroph or lithotroph, that something is using inorganic compound like methane, for example, or ammonia, like oxidizing something inorganic to produce energy. If something is organotroph, we are organotroph, so we are oxidizing organic compounds such as glucose to get reduction equivalents and to start electron transport chain. And there are third one, phototrophs, as the name says, those guys are able to use energy of light to generate energy, and those are first plants, so plants that they're doing photosynthesis, and microbes. We will talk about photosynthetic microbes, bacteria mostly, and also there are algae, but algae are between microbes and plants, macroalgae. So just basic things. Another terms that we should kind of follow is what means exergonic and endergonic reaction. So if something is exergonic, that means that reaction is releasing energy. If something is exergonic, you will see production of ATP and NADPH or NADH or FADH. So if you see reaction that says FAD goes to FADH2, that tells you it's exergonic reaction. It is creating an energy. Or ADP plus PI gives you ATP. The other thing how you can recognize exergonic reaction is by delta G sign, negative value. Just memorize it's completely opposite from that what you would expect. So negative delta G means energy productive reaction. Endergonic is something that is spending energy. So every time when you see that ATP and arrow gives you ADP plus PI, that would be endergonic. So all those transporters that we talked about yesterday, they are spending ATP, meaning that they need energy. Those reactions are endergonic. The same if you see, for example, oxidation of NADH to NAD plus, that's sign that's endergonic. And delta G is positive. Again, this is important for your general knowledge, and you need to know these terms to understand all further questions in units 7, 8, and 10. Not really 9, but 7, 8, and 10. So what means when substrate is oxidized? That means losing an electron. Then electron is jumping in electron transport chain through all those carriers. Energy is released, and proton is delivered across the membrane, and you have proton motive force formed. And that is the basic point of bioenergetic. Here are some examples of oxidation and reduction, and also terms that you need to understand to be able to read questions from the exam. So what is oxidant? So if something is an oxidant, that something is able to accept electrons. Just connect it with oxygen. Oxygen, oxidant. If something is a reductant, that is molecule that is able to donate electron. So reductant gets oxidized and oxidant gets reduced. So it can be a little confusing, but the way how I memorize it is oxygen. Oxygen is oxidant. And then I just know what's happening with oxygen in electron transport chain is getting reduced. That's where we are. Now, redox potential, or EO, is tendency of molecule to gain or lose electrons. That means that some molecules are good oxidants, so they're good in accepting electrons, such as oxygen, for example. But some molecules are great reductants, so they have tendency to release electrons. So that's where redox potential comes into the game. If something has a negative redox potential, that means it's a good electron donor. Hence, it can be oxidized, like NADH, for example. NADH can be oxidized to NAD plus, and that's the first step in electron transport chain. What means positive? So more positive redox potential are electron acceptors. That means they have affinity for electrons. That's why they are more positive. Like oxygen, for example, the one that is on the bottom, that one is good oxidant, has positive redox. And this is a Bible. Tower of Powers, apologies. It tells you how good couples some oxidant and reductants are. Again, what you can see, basically the best couple is on the top. Yes, so again, this is something that you probably saw already. Don't memorize it. Like if you get a question for this, you will be provided with this table. So nobody will ever ask you to memorize what are redox potentials. In summary, again, left-hand side, exergonic reactions are characteristic of catabolism. They have negative redox potential, and it's a top power tower. Endergonic reactions are in anabolism, positive redox potential, bottom power tower. And now ETC, or electron transport chain. So I will briefly go through this. It's already in, you have these notes also in the first review session, units 1 to 3. But this is basic of bioenergetic. So
quick question.
Yep.
When it comes to looking at things such as like electrons transferred and stuff, like in the slides, there's like how many electrons are transferred. Like, are we going to need to be able to do that for the final? Like, go through, I don't know if you can tell me.
So I will tell you when I look in details exams. I think you do need to know some basic things, like, for example, how many protons are needed for nitrogen fixation. So something like that, like not in details too much, but maybe in few reactions that Dr. Stein really emphasized, like, for example, nitrogen fixation. So you should be able to memorize that. But again, when I saw exam, but I didn't go through details. But I will, if there is something that you guys need to memorize, I will make sure to give you a hint. What's more important to focus on is how the process is happening, not how many electrons. Like that's my opinion. I don't think that Dr. Stein will be angry or take points if you make mistake in how many electrons are transferred, but you have a all process well explained. Does that answer?
Yeah, that's great. Thank you. There's just quite a few slides on it, so I wanted to make sure. Yeah, thank you.
Yeah, and ask me anything what you want. That's why I'm here. So anyway, back to the electrons. So where do electrons go? Electrons arrive to the complex 1. Complex 1 is NADH dehydrogenase. So what's happening then? NADH dehydrogenase is now oxidizing NADH in cytosol and creates NAD+. This is the first step, complex 1. In the same time, protons are translocated from the periplasm. So complex 1, NADH dehydrogenase, is a proton pump. Now, second in the line is complex 2. Complex 2 is not proton pump. Complex 2 is connected with TCA cycle, and we will mention that in a few minutes. Anyway, the second in line, complex 2, and then ubiquinone is linked to the complex 2. What's happening then? Q accepts electrons derived from NADH oxidations, and now you have QH2. So reduced quinone pool. So that's where electrons are. What's happening downstream in cell? In cell, you have TCA cycle that is happening apparently close to the membrane. And then in TCA cycle, you have succinate dehydrogenase that is oxidizing FADH to FAD+. Just that's simple TCA cycle, succinate to fumarate. But then those electrons from oxidation FADH to FAD are delivered to quinone pool. So that's basic points, but no proton pumping. Complex 2 is not proton pumping. Now, next step is electrons are moving toward cytochromes. Cytochromes are electron carriers. So electrons go to complex 3, cytochrome oxidoreductase. Cytochrome oxidoreductase acts on cytochrome B complex, and everything is in membrane. Cytochrome B complex now becomes reduced. That iron 2+ in cytochrome B, accepts electrons, becomes Fa3 plus. Oxidized. So you see, very easy to make mistake. So basically what's happening is oxidoreduction on cytochrome B, and this is done by cytochrome oxidoreductase. So once an electron comes to the cytochrome B, it comes that oxidoreductive reaction and protons are pumped. Terminal complex 4 is cytochrome AA3 or terminal oxidase. And those cytochromes are different from they're different from species to species. Not all of them, but definitely species specific. Now, this terminal oxidase holds oxygen, and basically it's like a clip that holds oxygen between iron and copper, and oxygen accepts electrons, becomes reduced to water, and protons are pumped. So complexes 1, 3, and 4 are proton pumps. They participate in generation of proton motive force, where complex 2 is just linked to the membrane. It does not pump protons. Now, again, I won't go in details what's happening when it's low oxygen. Basically all the same. So all these are the same like we went. You have NADH oxidized via NADH oxidoreductase, you have quinone pool, you have cytochromes, you have terminal oxidase. What is different is I believe you can see, terminal oxidase complex 4. Now it's cytochrome D instead of AA3. Because this complex has higher affinity for oxygen, because remember now you don't have enough oxygen, so you have to collect everything what you have. So it's just a difference in oxygen. And complex 4 does not pump protons. So everything is the same in minimum oxygen conditions, except terminal oxidase, and complex 4 in this case is not proton pump. Anaerobic respiration. What does that mean? It's kind of weird, because if something is anaerobic, means no oxygen, and we use the term respiration. So basically anaerobic respiration means that electron acceptor is something else but oxygen. So again, all is the same. Electron transport chain is the same through all complexes and quinone pool and cytochromes and everything. But again, we have now terminal, not oxidase this time. It's a reductase. And why? Because as electron acceptor, in this case, we don't have oxygen. It's anaerobic, 0 oxygen, but we can have some other thing, maybe nitrate or sulfate, or remind me, some metal can be also electron final acceptor. So that's difference, and also no proton pumping. So differences between like high oxygen conditions, low oxygen conditions, and 0 oxygen is in terminal enzyme and characteristics of complex 4. So in high oxygen conditions, you will have cytochrome AA3 terminal oxidase, where in minimal oxygen conditions, that will be cytochrome D. And in 0 oxygen conditions, it won't be oxidase, it would be reductase. And no proton pumping. Fermentation. Fermentation is, again, maybe more significant for 315 class, where we cover this in detail. But here, I will focus more on physiological part of fermentation. So what kind of reactions are in fermentation? So they're also redox reactions, just not so intensive as in electron transport chain, and way less energy is released. There won't be electron transport and no oxidative phosphorylation, no cytochromes, of course. What does that mean is that ATP is generated by substrate level phosphorylation. And you get way less energy. So just for comparison, and again, don't memorize this, if you have oxidative phosphorylation and full electron transport chain, I think you get around 36 ATPs, where with only glycolysis, which is part of fermentation, you get only 2 ATPs. So way less energy, but great products, like beer is one of them, milk, many other things. Anyway, what means homofermentation? Homofermentation means that at the end you have only one product. Heterofermentation, which we will cover later, means that you have multiple products. Homo, the same, hetero, different. Now, the starting molecule here is glucose, so that's where we explain everything. And glucose is activated via hexokinase. Again, I will just return you from to yesterday's talk when we talked about activation of transport, when we talked about transport, how glucose stands on membrane, held by some protein, where all those series of phosphor transfers happen, and then phosphate comes to the glucose, and pop, glucose is activated. So I'm just trying to connect what we talked yesterday, because it's microbial physiology and everything is connected. Now, this is hexokinase that transfers phosphor group to group yielding glucose 6-phosphate, and that's how you usually see glucose in organism. Rarely it's just glucose. Next enzyme is isomerase, which means enzyme that is just changing a structure of glucose 6-phosphate. So again, 6-carbon molecule just now is going to be fructose 6-phosphate. Just it looks different in space. Again, that's a chemistry, general organic chemistry, if I remember well. The key enzyme in fermentation, in glycolysis generally, is phosphofructokinase, or PFK. That's a rate limiting step, which means when PFK phosphorylates fructose 6-phosphate, you get molecule with 2-phosphate group, fructose 1,6-BP or bisphosphate. And from now on, everything is pretty much clear. So when you come to when organism comes to the point of building F16BP, no go back. So that's a sign that fermentation is continuous, or glycolysis, depending on which type of process you're talking about. Now, next step is having an aldolase that will split F16BP into the two different molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So, that's general glycolysis. In fermentation, in homofermentation, you have two same glyceraldehyde 3-phosphate molecules. Each of these molecules are further processed. And what's happening then is that GAP-DH, or glyceraldehyde 3-phosphate dehydrogenase, shortly GAP-DH, is oxidizing glyceraldehyde 3-phosphate, which means what? When something is oxidized, something else has to be reduced. So what cell will do, cell will reduce NAD+ to NADH. NADH is reduction equivalent that is needed for further synthesis. What the result of this oxidoreduction is 1.3 bisphosphoglycerate, or BPG. Now that's a 3-carbon molecule. And enzyme kinase is converting 1,3-BPG to 3-PG. Significant here is that you have one ATP molecule generated. In the next step, you have mutase that creates now 2 phosphoglycerate. So it just moves phosphate group from carbon 1 atom to from carbon 3 to carbon 2 atom. So you have 2-PG, and 2-PG is then converted to phosphoenol pyruvate via enolase. Phosphoenol pyruvate, you remember, high energy bond. Lastly, phosphoenol pyruvate is converted to pyruvate. Pyruvate kinase is leading this reaction, and this process is generating 1 ATP. So, since fructose 1,6-bisphosphate was split to 2 molecules, then we end up with 2 ATPs and 2 NADHs. In the next step, cell needs to regenerate oxidant, NAD+. So to do that, cell is using enzyme lactate dehydrogenase. What does that mean? Lactate dehydrogenase is now reducing pyruvate to lactate, and at the same time, it is oxidizing NADH to NAD, and that's regenerated oxidant. And product is lactic acid, or lactate. The same situation is happening with ethanol fermentation. Just instead of lactate dehydrogenase, you have ethanol dehydrogenase, or alcohol dehydrogenase, and the product is ethanol. hetero fermentation, slightly different. Again, starting molecule is again glucose, and you again have this activation via hexokinase. You have glucose 6-phosphate generated, but here is where is the difference now. Glucose 6-phosphate dehydrogenase is now oxidizes glucose 6-phosphate to 6-phosphogluconic acid. In the same time, NAD plus is reduced to NADH. So it's different. Over there in homofermentation, you had isomerase that converted G6P to F6P. Here, you have slightly different situation. Now, this 6-phosphogluconic acid is decarboxylated, and you get ribulose 5-phosphate. And again, NAD plus is reduced to NADH. Then you have ribulose 5-phosphate isomerized to xylulose 5-phosphate. And further on, reactions are completely different. Xylulose 5-phosphate is phosphorylated and split. So you get glyceraldehyde 3-phosphate and acetyl phosphate. Glyceraldehyde 3-phosphate, you already know that one can generate lactate, but acetyl phosphate is reduced to acetaldehyde, and NADH is, of course, oxidized. And then acetaldehyde will be reduced to ethanol, and again, NADH will be oxidized to regenerate an oxidant. So again, this is at the end of this, you have lactate and ethanol. In previous one, you have only lactate. And this is how it looks in picture. I think this picture is way better than just me talking. This is mini summary. Again, what you need to memorize. I mean, what you need to understand, let's say like that. So glucose oxidized to pyruvate. Pyruvate can be decarboxylated to acetyl coenzyme A. Acetyl coenzyme A shunt to TCA cycle. Pyruvate can be reduced to lactate, yielding NAD+. Pyruvate can be reduced to ethanol, yielding NAD+ regeneration. Pyruvate can also be oxidized, yielding acetate and formate via enzyme pyruvate formate lyase. The enzyme that is lysing, splitting molecule. Sorry about that, I cannot pronounce some words. Then basically, generally, oxidation of pyruvate through acetate results in production of hydrogen gas. What is important to understand is probably these three: pyruvate to acetyl coenzyme A, lactate, and ethanol. Central carbon metabolism. What does that mean? When we say central carbon metabolism, we refer to glycolysis, TCA cycle, and pentose phosphate or PP pathway. As a part of glycolysis, there are EMP and EDD variants. So in next few slides, you will see pretty much similar reactions that we already went through. So here is how glycolysis looks, EMP versus EDD. And again, this is just a picture. I will lead you through all these reactions in a few minutes. I mean, I think maybe better if you just see picture than if I just read slides. So in my field, in methanotrophy, EMP and EDD pathways are highly significant for gamma methanotrophs, and it's quite interesting how those bacteria are changing their physiological response and the preference of EMP and EDD pathway depending on the nutrient source. Pentose phosphate pathway, this one is interesting from the perspective that it's quite unique because it generates 5 carbon sugars. It generates material for nucleic acids, and it also generates some materials for like glyceraldehyde 3-phosphate, which can be shunt back to glycolysis to continue TCA cycle. And also you see that from many precursors from many products of pentose phosphate pathway are used for lipid generation. So this is one of main biosynthetic pathways. Glyoxalate cycle. This is unique for some bacteria, as far as I know. I can't remember whether there is an eukaryotic doing glyoxalate cycle. I usually know only about microbes, but I might be wrong. So in glyoxalate cycle, this basically modified TCA cycle, and here is how. So remember that acetyl coenzyme A that was produced by pyruvate decarboxylation. So in combination with oxaloacetate, acetyl coenzyme A gives citrate. Citrate is TCA molecule. Then logically you would have isocitrate, but then instead of having a regular TCA cycle where isocitrate would be converted to alpha ketoglutarate and further to succinyl coenzyme A, isocitrate is split by isocitrate lyase to glyoxalate and succinate. So succinate is regular part of TCA cycle. As we can see, cycle continues from succinate to fumarate, but glyoxalate is not. So glyoxalate interacts with acetyl coenzyme A, and this reaction is catalyzed by malate synthase. Then you get malate, which is again part of TCA cycle. So again, cycle continues, just you're skipping a few steps, alpha ketoglutarate and succinyl coenzyme A. And this would be all for unit 6. And now let's go to unit 7. Wait a second, what did I press? Okay, this one is hard. I mean, it's not so hard, just there will be many questions from here. Which is good. This is real physiology. This is like a real world. Ready? He has 45 slides, right? Oh God, we are dead. We are so dead. Anyway, I should get some water. Will you wait for me a minute just to get water, please? I'm coming fast. Sorry about that, guys. Anyway, study session unit 7. We talk about nitrogen assimilation and metals first. First thing that you need to understand and to probably memorize, well, not so much memorize, but understand that there are macronutrients and micronutrients. Macro stands for those that are needed in high amounts, like carbon, nitrogen, oxygen, phosphate, sulfur, hydrogen. I don't know why selenium is here, but apparently is. That's how literature says. And micronutrients are all those metals that are usually required most often for functioning of enzymes. Essential ions like sodium, magnesium, potassium, calcium, chlorine. There are some special function elements. I'm not sure really why cadmium is there, because cadmium is toxic as far as I know. And there are other unessential metabolites and non-metabolites. Non-metabolites are those gases, helium, argon, and neon. For you, it's important to understand macro and micronutrients and those ions that are usually regulating membrane functions. Very interesting microbe metal community are rocks, and Dr. Stein talked about that in unit 7. So what is important to know is that rocks generally represent iron oxides and manganese oxides. So they're solid, right? So when our planet became oxygenic, which happened thanks to the activity of cyanobacteria, so oxygenic event is actually a product of microbial physiology. Now, when planet became saturated with oxygen, iron and manganese oxidized and created solid formations such as rocks. So chemical processes that are involving microbes and metals are again oxidation and reduction. Now you probably you can see why it's important to understand unit 6 so you can understand everything later on. So oxidation of metals and reduction of metals, and iron and manganese are two metals that we are using as example. So both of them can be and oxidized and reduced. So in what's happening in manganese oxidation? So theoretically, bacterium should oxidize manganese and reduce oxygen. So manganese is highly redox positive. Some bacteria that form spore create a coat of manganese oxides around themselves. So those are probably what's important to memorize is that manganese oxides can protect bacteria as a spore. Specialized mangan oxidizing enzymes creating manganese oxides. So those are that solid black, like I think Lisa was talking about event that happened in the lab. So they look like a black sedimentation on lab dishes. So those are manganese oxides. So that protects bacterium. So in this way, bacterium is, yes, protected, but also bacterium created its own terminal electron acceptor. Why is that important? Well, when this organism sinks down in the highly redox negative zone, it might not be able to find anything to respire. So guess what? Now organism has its own manganese oxides, and that's now terminal electron acceptor. So basically, the purpose of the purpose of creating those manganese oxides around themselves for bacterium was to protect from unpleasant environment where there is nothing to respire. So that's pretty cool adaptation of bacteria and involves oxidation of metal. Iron oxidation is related to bacteria that contain specialized compartments or magnetosomes, so magnets. These bacteria are magnetotactic bacteria. So iron oxides are created by special enzymes and deposited in magnetosomes. That's how it functions. And these organisms use these iron oxides to detect oxygen gradient. So they actually identify a redox zone where there is enough oxygen and to use it as terminal electron acceptor, but they don't want too much oxygen because it can poison them. So iron oxides are also some type of adaptation for extreme conditions. So again, interesting fact is that searching for bacteria with magnetosomes is a strategy to find a life outside of our planet. So these were oxidations. What about reductions? So there are bacteria that respire metals. So as I mentioned a few minutes ago, oxygen is great for respiring, nitrate also, but there are some metals that could be respired, and those are, for example, manganese and iron. So oxidation of electron donor is unusual. So when we say oxidation of electron donor, the question is what is oxidized, like what is donating electrons. So electron acceptor will be metal, but what is oxidized is some organic fuel, glucose, for example, or anything else, like organic fuel, something that is good as electron donor. So oxidation of that fuel is happening with the purpose to produce NADH. Why? Because NADH is a beginner of electron transport chain. So when NADH runs through electron transport chain, that generates PMF. But now electron transport chain is not terminated at oxygen, it's actually terminated at mangan and iron oxides. So here is the question. You had that question on problem sets. Manganese and iron oxides are solid. So how bacterium respires solid structure? And discovery and answer for this question was resulted in creation of microbial fuel cell. And microbial fuel cell is very important for this exam. Now, how microbes respire solid materials located out of cell? For me, this was interesting. Like first time when I heard it, I was like, whoa, that really exists. Anyway, these solid materials are redox positive, so they're terminal electron acceptor. They can accept electrons. For example, is iron 3+. That's electron acceptor. The whole purpose of respiration is to bring electrons from donor to terminal electron acceptor, because while those electrons are moving, you see proton motive force. Protons are transferred across the membrane, and there is a proton motive force. Again, donor of electrons, that fuel that is oxidized, is inside the cell. So imagine bacterium is eating something organic, creates NADH, NADH beginner of electron transport chain. NADH oxidized, through all electron moves through all those electron carriers, proton motive force generating, and now bacterium has to deliver electrons to something, and that something is not in the cell, it is actually outside of the cell. So how bacterium does that? There are two options. One is electron shuttle, the other one is direct contact. So electron shuttle. Electrons now travel through cytoplasm, and they're coming to the outer membrane, and outer membrane has some extensions. These outer membrane extensions are nothing else but cytochromes. The reason is simple. Cytochromes are accepting electrons. Cytochromes are electron carriers. So that thing that accepts electrons has to be made of cytochromes. They're soluble, they accept electrons, and from cytochromes, electrons travel to humic acid. So humic acid is like a mediator. It's a shuttle molecule, how we call it. And humic acid actually delivers electrons outside to Fe3+. So this is respiration of iron, and it forms iron 2+. So it's like you have like extension of a membrane, like a pili. I mean, it's not pili, but that's how it looks. And then you have humic acid outside. It accepts electrons and immediately travels to the iron 3+. Direct contact. So, I mean, I guess it's pretty clear. What does that mean? So again, you will have electrons traveling through cytoplasm. There are also outer membrane extensions made of cytochrome. But now no humic acid. So it's basically like a direct contact, so no mediator. So the only difference is in that humic acid. And again, iron will be reduced from Fe3+ to Fe2+. And microbial fuel cell. I should write MFC, not MCF. Sorry about that. So microbial fuel cell contains two compartments. And again, there are various pictures available online. I didn't put any of them. I just wanted to lead you through it and to tell you what is important to know. So microbial fuel cell, again, two compartments. One is anoxic, the other one is oxic. Anoxic means no oxygen. Anoxic compartment contains an anode, which is made of iron oxide, Fe3+, the one that can accept electrons, where oxygen compartment contains cathode. So microbes are inoculated in anoxic compartment, and they are fed with glucose, regular fuel source. When microbe is oxidizing glucose, there will be many electrons generated. Those electrons are negative, of course, and they will travel to the anode that is made of iron 3+. Again, we know that iron 3+ is electron acceptor. When electron comes to the Fe3+, Fe3+ becomes reduced to Fe2+, and electrons are driven through anode. Those electrons now travel from anode to cathode in the oxygen compartment. Now, in oxygen compartment, electrons just continue to do their electron transport chain because there is oxygen there. They will reduce oxygen finally, and you will get H2O, so typical electron transport chain. So microbial fuel cell is nothing else but extended electron transport chain from anoxic to oxic compartment. The remaining energy is captured in oxic compartment and used to turn on some light bulbs. So it's important to memorize what is in anoxic, what is in oxic compartment, where electron transport chain begins, what is the donor, and where electron transport chain ends, and what is the acceptor. So in anoxic compartment, you have electron donor. Electron donor is glucose. So glucose is oxidized. Actually not glucose. Glucose is catabolized to get NADH, and then NADH is electron donor. First electron acceptor in an oxy compartment is iron 3+. But that's not the end. Electrons continue moving and they end up in oxic environment where terminal electron acceptor is oxygen. So it's important to know in which compartment is what.
Can I ask a question?
Yes, of course.
Is there a shuttle molecule in the microbial fuel cell?
I don't think so. I mean, I didn't find in Lisa's lectures, but I don't think it. I think it would be possible. It would be doable. What would you use as a shuttle molecule? Like something like a humic acid?
Yeah, or like, or maybe there's the cytochromes. I just was curious. But.
No, that's a good point. That's very good point. Now I'm just thinking. I mean, would you need that actually? Like, would you need shuttle molecule because electron is pretty much attracted by iron 3+ on anode?
But the electrons generated by oxidizing glucose, which is in the cytoplasm, right? And then it somehow get out. And
I see what you mean.
The anode.
Well, that's a good point, and it's possible that there is. I think it all depends which microbe you are using in an oxygen compartment. You can use microbe that has direct contact and does not have humic acid, but you can use microbe that has humic acid. So yes, I believe that would be a good point. So depends which type of microbe you are using in an oxygen. That's very good point, Carmen. Yes. So memorize, guys, this. So if you get that kind of question, yes. I mean, why not? It's possible, but remember that there are microbes that are using direct contact. So that's how you can get electrons out of the cell. Anyway, thank you, Carmen. And Geobacter. So again, 2 models. I won't go in details through these models, but what I want you to memorize is main point of microbial fuel cell, and Geobacter is try to memorize in these proteins like OMC, BEE. So because those are cytochromes that are important. You don't have to know exactly name, but know that they are cytochromes. There is another model. So again, different cytochromes. So look here, for Geobacter, you have BEE, OMC, B, OMC, E. I think I made a mistake. I wrote it right. Here you have OMCA, MTRC. So I wouldn't bother memorizing names, but what you need to know that those are cytochromes, because cytochromes are carrying electrons. Siderophores, metal homeostasis. What's the principle? So here we will have some connections from previous lectures. So what are siderophores? Those are molecules made by microbes to scavenge iron outside of the cell. I do have to emphasize, which is not mentioned in lectures, that siderophores are not unique for microbes. Plants also have phytosiderophores, but they're used for the same purpose, to take iron. Siderophores catch iron outside of the cell and drag it into the cell. Here is TONB. So TONB is a molecule that spans all inner outer membrane and creates proton motive force. Why is that important? Well, when siderophore catches iron, it will need to drag it into the cell. For that, you need some kind of energy, and that's where TONB will come into the game. So PMF created by TONB will drive that siderophore iron complex and get into the cytosol. Then iron is in cytoplasm and it's released through permease, ABC transporter, or efflux pump. Really depends what is connected with TONB. So TONB is kind of common. What is connected with can be permease, ABC transporter, efflux pump. Really depends. Important is to know that TONB creates proton motive force to drag in to drag in siderophore iron complex. Nutritional immunity, very, very significant. This one you need to understand. So what is nutritional immunity? So that's something that when host cell tries to fight against pathogen, nutritional immunity is created in collaboration with host immune system. All purpose is to use metals against microbes. So memorize nutritional immunity. Again, you have that in lecture, probably better written. There are two ways of nutritional immunity: metal sequestration or metals toxicity. So bacterium is using metals somehow to, oh my God. So we are using metals to protect from pathogens. Metal sequestration. So again, there are many molecules that are created by our immune cells, and the purpose of creation of these molecules is just to take metals from pathogens. So we want to starve pathogens for metals. Why? Because they need metals to function, and we don't want them to function in our cell. So I wrote here a few molecules that are found in immune cells. I wouldn't bother memorizing specifically name of the molecule. What is important to know is what those molecules are doing. So what they do, so our immune system creates this NGAL molecule, and what NGAL does is catches iron, chelation, that's what it means, and then bacteria can't get iron. Again, in macrophages, there is another molecule that does the same thing, but to iron and manganese. And also there is calprotectant in neutrophils. So all what they do is sequestering metal. They're catching a metal and prevent bacterium for grabbing it and functioning normally. What's the principle of metal sequestration? Again, this is very important component of the exam, especially calprotectant. So neutrophils generate calprotectant. That's a chelator. Calprotectant will chelate manganese and zinc. So bacterium needs both manganese and zinc to activate superoxide dismutase, or SOD. So these enzymes, SOD, Mn, or zinc, are used by bacteria to defend itself from superoxide radicals, because we produce superoxide radicals to kill them, and they have SOD enzymes and they defend themselves. Now, calprotectant took manganese and zinc. When no manganese and zinc, SOD cannot be active. So our superoxide radicals in neutrophils can now kill bacteria. Again, of course, microbes are way smarter than we will ever be. So they evolved some mechanisms and became resistant to metal sequestration. How is that functioning? So there is this MNOP, MNPH system. So this one is transporting manganese, and that basically helps bacterium to acquire additional manganese when host cell takes out manganese. So important to memorize here is that one of the mechanisms of resistance, or one of mechanisms that bacteria evolved to overcome our defenses, to just develop additional transporters. So yes, we will take manganese from them, but they will just develop more efficient system for the uptake. The other one, CYT system, binds manganese and drives into the cell. So it's pretty much the same. So you have transporters and receptors that are recognizing metals, and they're just increasing the flux of metals. So we do everything to take metals from them, but on the other hand, on the other side, they're just importing more and more. And the similar principle is for zinc. So this was for gram negative. For gram positive, again, there are additional transporters, additional systems for uptake. So it's good if you can memorize all these gene names, but I'm pretty sure that I'm forgetting this every year, and I won't even bother memorizing it. What I know always is that the way how bacterium overcame overcame our mechanism of metal sequestration is developing additional systems for metal uptake, so it can continue functioning normally even if our immune cells are sequestering metals from them. The other way of nutritional immunity is metal toxicity. Here is how that functions. So macrophages are immune cells that are able to produce copper, which has oxidation state +1, very toxic. So this state is highly toxic. So when macrophages are producing copper 1+, that kills microbes. So just metal toxicity. What's the problem here? Of course, microbes are smarter. So they evolved mechanisms to convert copper 1+ to copper 2+, and to protect from metal toxicity. One simple question: if copper is converted from 1+ to 2+, is it oxidation or reduction? Come on, I want to hear anyone.
Oxidation, isn't it?
Exactly. Yep. It's simple to know. So loses one electron, electrons are negative, it becomes more positive, right? That's how I memorize it. The principle of metal toxicity in macrophages. So macrophages is ingesting microbe inside the phagosome. What's happening in the same time is interferon gamma in the host cell induces expression of molecule CTR1, and CTR1 is nothing else but copper transporter. So cell is importing copper 1+. There is series of other transporters which you really don't have to memorize, but basically when microbe is inside of macrophages, that's initiated to trigger on the system for copper+ uptake. Then microbe suffers of oxidative damage and dies inside the phagosome. But again, metals evolve to fight against, microbes evolve to fight against metals toxicity. You probably already now know that these structures are efflux pumps, transporters. We give them metal, they get rid of metal. They're just effluxing. It can be P-type ATPase, R&D family, ABC transporters. Those are all secondary transporters that we talk about because microbe just wants to push toxin out of cell. Copper homeostasis is very important. Make sure you understand all 4 of them and to memorize them. So copper is significant and it's important for many reasons. First of all, it is essential nutrient, micronutrient, but it can be also toxic. That means that regulation of copper uptake has to be very tightly controlled. There are 4 ways how microbe controls copper, and we'll go through each of them. First one is copper efflux pump. Second one, P-type ATPase. 3, periplasmic accumulation of copper and oxidation of copper to copper 2+. Let's go through efflux pumps. So when copper enters in microbial cell, of course, it's trying to kill it to cause oxidative damages. Microbe has this protein Cas-A in the membrane. Cas-A is just recognizing copper, and sometimes there are some chaperones F and B, C that are helping this delivery. And other possibilities, direct delivery from Cas-A to Cas-B, spanning system in the membrane. So this is the first way how bacterium regulates copper. So when there is a copper plus, Cas-A recognize it and shuts and transported it out. P-types ATPases, as you know, transporters that are hydrolyzing ATP to gain energy. There are 2 types of transporters, COP-A and COP-B. They are under the same promoter, and activity of each is tightly regulated by the level of copper. This one you need to understand. Here is how regulation of these 2 genes in operon functions. Promoter is itself regulated by copper. And as a part of the promoter, there is a place for binding of 2 types of molecules, repressor and anti-repressor, Y and Z respectively. When there is no copper plus, so no danger in the system, COP A and COP B will proceed. So both of them will be expressed. Repressor Y won't be active. Again, both of those are expressed but basal level, so nothing special. When copper level is moderate, so not a big deal, not urgent, at both sides of membrane, copper binds COP-Y repressor. Now, why this repressor COP-Y copper occupies promoter and blocks the expression of A and B. So moderate level of copper blocks the expression of COP A and B. When copper level increases greatly, so you have high copper, copper plus binds to anti-repressor, COP Z. Z copper now binds to Y copper and prevents its binding to the promoter. When repressor cannot bind, expression will proceed. Depending on the copper level, you see expression of A or B type. If it's lack of copper, you will see A for the uptake. If there is too much copper, you will see B for the efflux. So this one, repressors, anti-repressors, that's what you would want to memorize. Third one, periplasmic accumulation. There are copper binding proteins like BDA, so they are just binding, chelating copper from outside of cell, like in periplasmic space, really nothing special, no special mechanism. And the fourth one is oxidation from copper +1 to copper +2, because +1 is toxic, +2 is not toxic. So when cell imports copper +1+, that forms free radicals, causes oxidative damage. And inside the cell, bacteria developed some multi-copper oxidase. So that's all, just an enzyme that is oxidizing copper 1+ to 2+. Once when you have copper 2+, that's easily exported through Cas system. And two-component regulatory system, again, always very important. We all love this two-component regulatory system. So here is how it functions. So Cas-S, this system that we talked about, that is oxidizing copper. This is histidine kinase. The word tells you everything. Histidine kinase has autophosphorylative activity. So when copper is high, Cas-S is doing autophosphorylation and creates Cas-S phosphate. So histidine kinase phosphorylation happens on histidine. This phosphorylated Cas-S passes further phosphate group to response regulator, Cas-R, and now you have Cas-R phosphate. Cas-R phosphate activates expression of transporter to remove copper. So it's simple two-component regulatory system. Cas-R is transcription factor. Nitrogen assimilation. That will be, so you will have here nitrogen assimilation, but you will also have this in the final unit 10. So memorize it. There are memorize processes. So what is nitrogen assimilation? First of all, that's unique for microbes. Bacteria are only organisms that can fix microbes, that can fix nitrogen, sorry. So nitrogen assimilation or fixation of atmospheric nitrogen is unique to the bacteria, but nitrogen assimilation also goes through assimilative reduction, and here is how. First, fixation of atmospheric nitrogen. Again, I won't read everything here, I won't talk about everything here. What I want you to know that fixation of nitrogen is energy extensive process. Know how many ATPs, and I would memorize how many protons are used, and know that what is main product of reaction. So this sense that nitrogen fixation is done by denitrogenase enzyme. What I want you to know is that nitrogenase enzyme is highly sensitive to oxygen, and this enzyme requires huge amount of energy. So it's energy expensive process. And again, I won't go through all of these details. I mean, it's just simple how nitrogen fixation functions. I would memorize that equation, or at least I would know what is the main product of nitrogen fixation. Main products are ammonia. So make sure to know this. NH3 is ammonia. If it's NH4 plus, that's ammonium. So that's something that we need to know. So ammonia and hydrogen are main products. These are some basic characteristics that I already mentioned. Energetically expensive, sensitive to oxygen, and nitrogen fixators are in some symbiotic relationship with leguminous plants. Carmen, I believe you are expert in symbiosis and you probably know more about this symbiotic relationship. But what they do, I think I'm right? So what they do, plants are providing protection from oxygen for nitrogenase, and nitrogen fixators are fixing nitrogen and provide ammonia for plants. So it's symbiotic relationship. Am I right? Yeah. Okay. I'm okay. I'm not expert in symbiotic relationships, but this one I kind of know. So, but I have to double check myself. So as I wrote here, that's all fine. Assimilative reduction. What does that mean? So there are some enzymes in microbes that are able to reduce NO3 or nitrate to nitrite. So again, more talks about this will be in unit 10 tomorrow. So these enzymes that can reduce nitrate to ammonia, which is biologically available form of nitrogen. So when we say that something is assimilative reduction, for example, in methanotrophy, nitrate, NO3 minus is methanotrophs can grow using nitrate. So the question that we usually get is, but how bacterium grows with nitrate, because like it's not biologically available form? Well, but it has enzyme to do assimilative reduction, assimilative reductase. These enzymes are grouped in categories NAP or NAR, depending on where they are located. So, but they do the same thing. So basically, how that functions is that cell expresses transporter for nitrate. When you give them nitrate, they will express transporter for nitrate. That transporter brings in molecule in the cell, periplasm or cytoplasm. If bacterium using NAP enzymes, then nitrate will be in periplasm. If bacterium uses NAR enzymes, nitrate will be in cytoplasm. Now, nitrite, NO3 minus, is reduced to nitrite via nitrite reductase, and nitrite is reduced to ammonia via nitrite reductase. So it's just a process from NO3 minus to NO2 minus and to NH3. In summary, again, inorganic nitrogen can be in form of N2 or dinitrogen, atmospheric nitrogen, and nitrate. Dinitrogen is fixed by nitrogenase to get ammonia. Nitrate is converted by assimilative reduction by nitrate and nitrite reductase to ammonia. All point is to get ammonia. Why? Because ammonia is biologically available form of nitrogen. How we know that? Well, if we think about amino acids, you have that NH2 group. So if you have available ammonia, that just goes and attach and you get amino acid. Yeah, so ammonia is biologically available form of nitrogen that is further incorporated into biomass through amino acids usually, and that's universal. There are two mechanisms of control of ammonia assimilation: glutamate dehydrogenase or GDH and glutamine synthetase glutamate synthetase GS-GOGAT. This is very important for exam. Regulation of these two systems, I would really be happy if you memorize and know. And I will make sure to explain well. GS-GOGAT system, sorry. Oh. Here is what's happening. Once ammonia is in the cell, at neutral pH, it becomes ammonium. So this is a difference. And Dr. Stein insists on that. So NH3 ammonia, NH4 plus ammonium, it's ion. NH3 is gas, NH4 plus is an ion. In the cell, because it's neutral, you have NH4 plus. Ammonium is at the same oxidation state for organic compounds, and it's easy to assemble it to other forms. The first step is to build the universal nitrogen donor. And when somebody asks you what is universal nitrogen donor, you will say it's a glutamate. Not glutamate, it's a glutamate. So you have ammonia entering the cell, ammonia becomes ammonium at neutral pH, and now that ammonium is used to build universal nitrogen donor glutamate. How to do that? Well, the cell does that by taking alpha ketoglutarate from TCA cycle and connecting this ammonium to alpha ketoglutarate. So alpha ketoglutarate plus ammonium, you get glutamate. Now you're all set. Universal nitrogen donor glutamate is created. Reaction between alpha ketoglutarate and ammonia is catalyzed by GDH. And that's, if you remember, glutamate dehydrogenase. When you see dehydrogenase, you immediately connect it with oxidoreduction.
Quick question.
Yep.
You say that it interacts with ammonia to build alpha ketoglutarate, but in the cell, isn't it ammonium? So is there
ammonium. Sorry if I pronounce it wrong. I'm sorry. Yeah, it's ammonium.
Okay, I just want to make sure. Okay,
yeah.
So
that's very important.
Step 4 happens with ammonium.
Yeah. So again, if I pronounce it wrong, please correct me. I'm constantly repeating it just so I don't confuse you. But just in the cell, ammonia becomes ammonium, and ammonium ion interacts with alpha ketoglutarate.
Okay, great.
Good.
Thank you.
And of course, just tell me everything what you notice. That's like all confusions have to be avoided this time, especially when we talk about dysregulation. Oh, I have to know this very well. If I don't know this at 3 a.m., I'm dead. So I need to know this, and I do. So anyway, now we have built glutamate, and you have spending of one reduction equivalent, NADPH oxidized to NAD. NADP should be NADP plus. The result is glutamate. Quick question, what kind of reaction is this, endergonic or hexagonic? Come on, tell me, I won't move forward. Is it, when NADPH is oxidized to NADP plus, is it hexagonic or endergonic? Who knows? Carmen, do you know?
I was letting someone else answer.
Just say it.
Well, this is a simulation, so I'm gonna guess endergonic.
You are right. Yes. You have a spending. No guessing. You are right. You are. I know. I do the same thing when I say, "Well, I guess," and then my boss would correct me. And we are not guessing. We are scientists. So I was like, "Yeah, okay." Many times I guess. So anyway, what's happening further? If concentration of ammonium, you see my mistake, so please remember ammonium, or actually no, this is ammonia. If concentration of ammonia is very low, so no enough ammonia outside of the cell, additional enzyme must be involved to build second most important nitrogen donor, glutamine. So when there is no ammonia outside the cell, there will be low ammonium inside the cell. All what you need to know is that even at neutral pH, ammonia becomes ammonium. Glutamate synthetase is an enzyme that is activating if there is a lack of ammonia, like not enough nitrogen, not enough biologically available form of nitrogen. So glutamine synthetase takes that glutamate that was built and adds one additional ammonia, which spends 1 ATP. This results in glutamine. Glutamine now reacts with alpha-ketoglutarate in reaction catalyzed by second enzyme GOGATE. That's why we call this GS-GOGATE system. And this is again endergonic reaction that spends NADH, and you see as a result 2 molecules of glutamate. So all purpose of GS-GOGATE is to build more glutamate because that's universal nitrogen donor. Now. Ammonia is assimilated through what you need to know. So ammonia is assimilated through GDH or glutamate dehydrogenase and GS-GOGAT system. These two systems creating 2 main nitrogen donors, glutamate and glutamine. First nitrogen, or the most important, is glutamate. So when you have enough nitrogen, enough biologically available nitrogen, you won't see too much glutamine. When you see a lot of glutamine, that means that no much ammonia is available. From glutamate and glutamine, all other amino acids are synthesized through different reactions of transamination. For example, pyruvate can take amino group from glutamate, and in reaction catalyzed by transaminase, it creates alanine. Then in reaction of deamination, glutamine can lose one amino group and becomes glutamate. Sorry, I made a mistake. So glutamine loses one amino group and becomes glutamate. If cell needs to create D-alanine for cell wall to build D-alamotive, in reaction of racemization, L-alanine will be converted to D-alanine. So you will get, so basically, here is connection. Pyruvate takes amino group from glutamate. This reaction is catalyzed by pyruvate transaminase, cell gets alanine. That's L-alanine, then racemization comes and you get D-alanine. Again, some summary, amino acids are a result of amination of carbon skeleton. So you have a bunch of carbon skeleton molecules and adding up amino group creates amino acids. Glutamate and glutamine, universal nitrogen donors for all other amino acids. Glutamate and glutamine donate amino groups to other amino acids. Amino group can be removed, which is deamination, or it can be reorientated, which is racemization. And again, ammonia is the gas, ammonium is the ion. Yep. And this is now regulation of these two systems, GDH and GS-GOGAT. So glutamate dehydrogenates activates when nitrogen levels in the cell are normal. So this system is constitutively active because cell always needs nitrogen. GS-GOGAT is activating when nitrogen levels are lower. So the way how you will know that cell is experiencing nitrogen starvation is increasing glutamine. Glutamine has 2 amino groups. So cell is starving for nitrogen, needs more nitrogen. Let's build glutamate. It has let's build glutamine. It has 2 amino groups. And regulation of GS-GOGAT is happening at 2 levels, transcription and enzymatic level. Both of them very significant. So what's happening at high level of glutamine? So there is a protein, GLND, that senses intracellular concentration of glutamine and alpha-ketoglutarate. When glutamine is high, alpha-ketoglutarate is low. GLND is occupied with glutamine. So basically glutamine is all around this molecule. Now, GLND activates uridyl removal or URA's activity. So these activities are very important. The result is deuridylated GLNB. So you have GLND that has UR or UT activity. Downstream is GLNB. When glutamine is high, you have deuridylated GLNB or naked GLNB. Naked GLNB interacts with GLNAE. Now GLNE activates ATA's activity and adds 12 AMP groups to GS subunit. What does that mean? Is glutamine synthesis becomes inactivated because there is enough glutamine. So key point, make yourself key points. High glutamine means low alpha ketoglutarate. This means URA's activity of GLND. This means deuridylated GLNB. This means ATA's activity of GLNE. This means GS occupied with AMPs, aka inactive. The reason for that is there is enough glutamine, no need to synthesize it more. When glutamine is low, GLND is sensing it. Know who is sensing. It's important which molecule is sensing level of glutamine. When glutamine is low, that means alpha ketoglutarate is high. GLND is not occupied by glutamine. There is no glutamine. Now, GLND without glutamine activates its UT activity, so adding uridyl group. The result is downstream molecule B, GLNB, with uridyl group. Now, uridylated GLNB interacts with GLNE. That means that GLNE activates adenyl removal activity and removes AMP groups from GC. So no AMP glutamine synthetase active to create more glutamine. Question: if glutamine synthetase is active, that means that it is making glutamine, right? What does that mean? Is cell starving for nitrogen or cell has enough nitrogen? And give me any thoughts. What do you think? Carmen, you can say it. I know you have thoughts. So please. Or Jennifer. Come on, I want to hear grad students.
I have no thoughts. My thoughts.
Would it mean that...
Let's go again. Let's go again.
Would it mean that the cell is starving for
exactly.
nitrogen?
Yeah. So glutamine, when glutamine synthetase is active, that means cell doesn't have enough nitrogen. You need glutamine. And think about that. Low glutamine, high alpha ketoglutarate. So low glutamine, here is the thing. Low glutamine, high alpha ketoglutarate, active GS. High glutamine, low alpha ketoglutarate, inactive GS.
And we can remember that because glutamine is the universal nitrogen.
Glutamate is the first one.
Right. Glutamate, sorry. Yeah.
Yep. Yeah. And glutamine is second most important.
Right.
So the way how I memorize it is because I did one, like my recent metabolomic analysis resulted in a high level of glutamine when cells are growing on nitrate, which told me very clearly that cells don't feel good about nitrate. They're starving. That's why they're building a lot of glutamine. But they still were fine. Anyway, mechanism of regulation at a transcriptional level. So we have stringent response and two-component regulation. So here is what's happening. In nitrogen starvation conditions, there will be a lack of amino acids, right? That's a stringent response. The lack of amino acid causes that stringent response, and the start of stringent response resumes activation of some RNA polymerases with alternative sigma factors like sigma 54. So no classic sigma 70, now we have activation of sigma 54. So lack of amino acid, stringent response, activation of RNA polymerase with sigma 54. Those genes that sigma 54 regulates are genes for biosynthesis of amino acids. At low glutamine and high alpha ketoglutarate. So this is now two-component system. NTRB is nothing else but sensory histidine kinase, which senses nitrogen level. Low glutamine, high alpha ketoglutarate. When glutamine is low and alpha ketoglutarate is high, NTRB becomes autophosphorylated at histidine. The resulting phosphorylated NTRB passes phosphate group to response regulator, which is NTRC, NTRCP is now active and turns on the expression of genes for glutamine synthetase. Because cell needs more glutamine, glutamine is starving. When it's high glutamine and low alpha ketoglutarate, again, sensory kinase. So, oops, sorry, I messed up something. Again, NTRB sensory kinase senses nitrogen level in the cell. So glutamine is high, so cell has enough nitrogen. Alpha ketoglutarate is low, and now no phosphorylation of sensory kinase. When no phosphorylation, NTRB binds to GLNB, remember from a previous regulation, and that prevents further phosphorylation. When no phosphorylated NTRB, there will be no phosphorylated NTRC. That means there will be no activation of glutamine synthesis of GS expression genes. This is just a detailed description of operon. I don't think you should memorize detailed description, but you do need to know regulation of operon. So there are two promoters, promoter 1 and 2. Low alpha ketoglutarate and high glutamine signals the activity of normal sigma factors like housekeeping genes. So basal expression of glutamine synthesis is maintained. But when there is high alpha ketoglutarate and low glutamine, so starvation, the activity of sigma 54 polymerase is triggered, and we have activation of glutamine synthetase. And this is just a summary. Integration of enzymatic and transcriptomic regulation when it's high level of nitrogen. So, So high glutamine and low ketoglutarate. So GLND turns on UR activity, uridyl removal. Then GLND removes UMP groups from GLNB. So GLNB is deuridylated. Deuridylated or naked GLNB interacts with GLNE. GLNE now, because of this naked interaction, activates ATA's activity. What's happening is GLNA adds CMP group and inactivates glutamine synthetase. In the same time, this naked deuridylated GLNB binds to the NTRB kinase and inactivates it. Naked GLNB stimulates phosphatase activity on NTRB. So all what you want, non-phosphorylated sensory kinase. When it's not phosphorylated, NTRB cannot phosphorylate response regulator NTRC, no NTRCP, there will be no transcription of GS genes. And now low level of nitrogen. So now we need, so here was high, so no transcription of glutamine synthetase. Now when there is a low level of nitrogen, we want glutamine synthetase. So low glutamine, high alpha ketoglutarate. GLND first player. Now it's UT activity. So adding uridyl groups. So GLND adds uridyl groups to GLNB. Now GLNB is not naked anymore. Now uridylated GLNB interacts with GLNE, and now opposite, GLNE activates adenyl removal activities. So all adenyl groups are removed from GS and GS is activated to make more glutamine. In the same time, uridylated GLNB cannot bind to NTRB because only when it's naked GLNB, it can bind to sensory kinase. Now, because it's uridylated, that blocks binding. That means that now NTRB kinase can do autophosphorylation and further phosphorylations on NTRC. NTRC is response regulator, two-component system, and NTRC phosphorylated activates transcription of genes for glutamine synthetase. And this is all for now, and it's releasing energy. I forgot, Brittany, what was the question? Yes, yeah, now I know, I see, yeah.
I have a question just about the kind of detail. Like you've used a lot of gene names like GLN, DBE, and then Dr. Stein was using more, like she used your UTAs, UR, ATAs. I mean, she gave us a table like of all these names, but then when she was explaining it all, she didn't use them as much. So what should we know?
I mean, I memorize it in my way. She memorizes in her way. I believe that you should do what's easier for you. Like for me, it's easier to memorize gene names, but I believe that Dr. Stein's system, UTAs and URAs, is much better because you don't have to memorize so many letters. So I believe it would be better to just memorize UT, UR, AT, AR. Okay. I mean, that's if it's easier for you. Like
they're more functional names than they are just sort of gene names.
Exactly. I mean, that's probably better it's easier to memorize. If you memorize gene name, you will definitely memorize, or enzyme name, you will definitely memorize what kind of activity it has. So for me, it's easier. The way how I memorize it, I arrange in my head. I have photographic memory, and then I arrange like DBE. That's how I memorize. That's how it's easier for me, which doesn't have to be for all people. Whatever is easier for you, as long as you know when it's uridyl removal activity, when it's uridyl addition, when it's adenylation or adenyl removal. That's what you need to know.
I think also based off what Dr. Stein was saying, like in lecture and stuff, like even she forgets a lot of the gene names and that kind of thing. And so I think that like, since this is open book, like I'm more going to focus on like kind of what you said with like the when each thing is happening, and then like refer to the actual gene name when I like summarize like each process. You know what I mean? Like exactly. That's going to be my studying level. So I'm going to know it at surface level and then being able to refer quickly to get the gene name.
That's exactly how you should do. Like, again, I have weird system of studying and usually way more complicated than normal people, but that's just me. So as long as you understand, like questions that you can get will be GLN or activated UR activity of GLNB. What does that mean? High nitrogen or low nitrogen? That kind of questions. So that's what you need to understand. And we will go through that once when I go through. Oh, Lucy asked something. Okay, no, that's what's okay. So anyway, that would be all for me. So you guys are free to go. I will convert this to YouTube file and we are good. Oof, I think I lost my breath a little bit.
Thank you. Oh. That long session.
Yes, I didn't talk so much, even like maybe if I'm arguing with my husband, yeah, but never again. Never in any other opportunity. I don't talk too much.
Yeah, I was focusing on this exam, and now with the announcement yesterday, I'm really worried I'll be kicked out of the lab here.
No, why would anyone kick you off the lab?
Well,
no.
Because I'm not essential, right, to here. So I'm like, oh my gosh, I have to do like lab work. So now I'm trying to do some lab work.
Whoa, that's not going to happen. Like, they won't kick us out. We are graduate students, right? We are safe.
Well, they did in the springtime, didn't they?
So I know what you mean. Okay, I thought about firing. Okay.
Oh, no. I just am worried I'll get an email today saying you have till Saturday to finish all your to get everything out of the lab. Anyway.
No, boy. Okay. Anyway, no worries, guys. We're good. You are all good to go. I will have to stay. So that's it. Bye.
All right. Thanks, Marina.
Of course. See you guys. Hi.